Hello and welcome to the Badger Talks podcast, the podcast that shares interviews with experts from the University of Wisconsin-Madison community about their work, research, and a little bit about what they're like as people, too. I'm your host, Buzz Kemper. Today, we'll be talking about genome editing, and I'm happy to have as my guest Dr. Krishanu Saha, Professor of Biomedical Engineering at Wisconsin Institute for Discovery. Krishanu Saha, thank you so much for coming in and doing this. I think this is a topic that's very deep, and hopefully we can make some sense out of it. You are involved in genome editing, correct?
Yes.
And why is this done, and what is the end goal of being able to edit genomes?
First of all, Buzz, thank you very much for inviting me here. It's a pleasure to be here. The goal of genome editing is really to be able to change the letters of DNA in a way that's beneficial. For us, we're interested in making changes that help with medicine. So one of the most exciting projects we have is to correct misspellings in the genome that lead to blindness. And these are inherited mutations where as little as one base in 3 billion in our genome are different than what you would find in everyone else, really. And that one base leads to progressive vision loss. And so we are using genome editing to try to change that misspelling, to correct the spelling, to provide therapeutic benefits for vision.
Wow. So where do these misspellings come from? You said they are genetic, they're passed down, but at some point they had to be just accidentally introduced, right? They weren't passed down for back into infinity, or were they?
It's a great question. I think there's an evolutionary story for many of the mutations that we see in patients. So there's actually two types of inherited diseases that we are attempting to fix at the genetic level and provide therapy. One is a recessive trait, which means that the parents of the patient had one of their two copies of the gene with that misspelling. Okay. And then it happened that the two copies that had the misspelling came together to provide a recessive trait and disease in the patient. And so usually the parents are unaffected, and the family lives, a kind of normal life, not knowing that they are carrying this mutation, but then see the disease in their child. So in some cases, this is kind of a pediatric diagnosis, and there's a very engaged patient community that is looking for new therapeutic options. In other cases, it's a dominant disease where one copy would give you the disease, and it doesn't matter if the other copy is normal or not. And the inheritance pattern there is that usually you have a parent that gave you that copy and they had an experience with the disease. Okay. And so they're actually very different kind of lived experiences of where these come, of what these different mutations might have in a patient's life and a family. Back to your original question, why would this happen in some ways, right?
Yeah.
So I don't know the full evolutionary story of the particular mutations that we are targeting in these patients and on why they might have arose. It can be bad luck that there is an error in the kind of copying of that part of the genome during reproduction, and that gets passed down. It could be that change in the genome could have some evolutionary benefit, potentially in a different organ in the body, or is expressed differently in a different environment or context. So the sickle cell mutation is sometimes called the sickle cell trait because it can be protective against malaria and other forms of infectious disease. So why we have these sometimes is a pretty complex and interesting story. Our lab is more focused on the cases that clinicians come to us and say, we've seen patients with these changes, are there new ways to go in and precisely fix that misspelling?
Okay. Wow. I have so many questions based on what you just said. So first of all, let's make sure I'm understanding the idea of dominant versus non-dominant mutations. I think what I heard you say was that in a dominant mutation, the parent and the child will both suffer the ill effect. But if it's not dominant, the parent may have no effect, but it may impact the offspring. Is that correct?
Yeah, that's exactly right.
Wow.
Yeah.
So why is that? How can a misspelling leave the parent unscathed, but then affect the offspring? How does that work?
It typically has to do with additive effects of the two copies of our genome. So we have a paternal and a maternal copy,
right?
And sometimes half of the levels of a protein that comes from the genome is good enough, and the cells in our tissues and we function fine, even though the other copy is different and doesn't and has the mutation and doesn't make the protein. So that's one reason why the parents might be okay, but if both copies have issues, and then you go from 50% of, let's say, a normal protein level down to 0, then you start to see effects.
Okay.
So that's one way in which the parents and the child may have different phenotypes and traits and effects. Okay. The other issue could be that the other parts of the genome matter. So it's not that we have all function dependent on one gene. Having compensatory changes in other parts of the genome that rescue the effects of one misspelling very well could be in the parents, which have a similar but different genome than the child, right? So those are all the ways in which the same mutation in a family may come about and have different traits in different generations.
You said that sometimes there are mutations that will have a negative effect in one part of the body. Maybe vision loss is one example, but it may have some benefit in some other part of the body. How do you balance that? First of all, how do you even find that out? But then how do you balance that? Is it worth correcting the bad thing, but then losing the good thing? That seems like that's got to be a tough dilemma for you.
Yeah, I think it's a very interesting and relevant question for sure. We're pretty careful when we think about exactly our disease target, and then downstream of that, what is the exact base and genetic target we are attempting to edit. And I want to emphasize that when we are trying to do this genome editing, we end up changing only a small portion of the eye. So it's really the changes that we're making are pretty precise and localized, arguably to less than a millimeter squared. So that's the tip of your fingernail or so. And we are not ideally trying to change any other part of the patient's genome.
Okay.
And I think we have a great localized target that has a big functional effect, because that part of the eye is so important for vision. There are other genome editing drugs that are injected into the bloodstream systemically. And there you do expect editing to happen in multiple tissues. And if you're able to pick the right gene that's only expressed, let's say, predominantly in the liver and doesn't really do much anywhere else, it's okay if it ends up editing parts of the kidney, because that gene is not doing much there.
Okay.
but then the therapeutic effect is happening in the liver, and that's where you're doing most of your editing. These are all important questions that the Food and Drug Administration asks before we get into actually trying and trying these candidates in human trials and clinical trials. There is another path that some investigators have explored, at least in animals, and then a few controversial experiments in humans where there is editing at the embryonic stage. And in that case, all of the cells of the body would carry the edit, including the germ cells, the sperm and egg that would be passed down potentially to subsequent generations.
Okay.
And so those are the types of applications that we and many in the community have thought very carefully about, and in some cases call for moratoria or limits on that type of work. My lab is not engaging in that. We are very much busy full-time trying to edit a small piece of the eye. But we also have, some projects that take out cells like blood or bone marrow and then try to edit those cells and then put them back in to the patient. And in those cases, we are editing larger portions of the body, arguably the whole entire blood system. And that has led to really encouraging and fascinating results in patients from work in the field. And so the first approved drug, Casgevy from Vertex Pharmaceuticals, was able to treat sickle cell disease using that approach.
Wow. So I was going to ask you then about victories. How is the research on respelling the this genome that affects the eyesight? How you're having success with it? How is that going?
So we're in preclinical stages. It's going extremely well. This is a large effort supported by the National Institutes for Health, where we are in the stages of engaging with the FDA, and we have a meeting with them upcoming here in a month or two about an important phase of our development to get their review of our data and approach on whether it's safe for moving forward into larger animals and then eventually into patients. And so the support that we have from the National Institutes for Health, specifically the Somatic Cell Genome Editing Consortium, is designed to put in a investigational new drug applications to start a clinical trial in 2028. Which sounds a little bit far away, but it is 2025.
Yeah, it's not that far.
It does take a large team. So we're fortunate to have clinicians, chemists, bioethicists even, and the patient community also contribute to how we are designing the drug itself, how we would conduct the clinical trial, and how we would try to make this type of approach into something that could be more of a platform, such that it's not so specific to one mutation. That if a patient comes in, let's say, with a different mutation, we can quickly use the same approach to develop a safe and effective drug. And so that in itself, that latter vision of trying to make CRISPR, which is the technique of genome editing that we work on, into a platform is really a major goal for that consortium that I mentioned at NIH. It's been supported through really a national effort for about 6 to 7 years now, and we hope that it ends up with real important and ambitious clinical programs at the end of this support. There's been amazing tools and other new types of editing strategies that have been developed through that effort, which in itself is a big success. And we've learned a lot about what genome editing can do in model systems. So we engineer little bits of the retina in a dish that look like the eye,
Nice.
and that's our model to test some of these genome editing drugs. And there's other that are making like mini brains, mini hearts, mini livers in really kind of simplified ways that we can interrogate and examine at high resolution, such that before we go into patients, we have a very good idea of the limitations of what we're potentially exposing these trial participants to.
Yeah, and speaking of limitations, this seems like the more you talk, the more I realize that there's potential for this and this. How big is this? Does this have the potential of being able to cure all kinds of diseases and maybe even go beyond curing problems, but actually strengthen the things about the human body that now tend to be more vulnerable? I mean, this is the potential for this as huge as I think it is?
I think so. I mean,
Yeah.
I'm very optimistic, really excited about where we are with the science and the engineering and the technology. I do think the investments that we've made in basic science, in kind of pre-competitive technologies, engineering, and the fact that the community has shared a lot of their advances openly, data sets, materials, maps, genetic maps of how you would make an editor, have really accelerated progress. There's been even a set of AI approaches to make new editors that companies and academics are now starting to see and play with, at least in these types of model systems. So it is really exciting in terms of the capabilities we have.
Yeah.
I think the potential to rewrite different parts of our body is really interesting.
Yeah.
And as a bioengineer, it brings new capabilities of sensing disease perhaps before it happens in the body.
Right.
Being able to put in genetic circuits that respond to injuries, infectious agents, cancer, in really new ways that we haven't imagined really in some ways. So that in itself is, I think, really exciting for me and my trainees and my lab members. And as the field thinks about being able to take out parts of the body like the blood system or a piece of skin and then maybe put it back into a patient. Changing the genome is fundamental to being able to put in new functionality into parts of the body. So I'm pretty I guess, careful about how I think about where we should go.
Of course, there are all kinds of, ethical and other questions involved, but the potential is amazing.
Right, exactly.
Yeah. As I'm listening to you talk, this could be like do what a meteorologist does, be able to look at genomes and be able to predict what is going to happen in the future for that person. This spelling of this genome is likely to lead to X. Is it predictive? Am I correct in that?
I think you're close, and there's certainly...
I'll settle for close. Yeah. So where am I wrong?
I mean, I don't want to be a genetic determinist that you are what your genome. Of course, it gets expressed. Right. It matters how you live and the types of experiences you had. So,
of course,
it is.
I'm talking about tendencies, not certainties.
Yes. So, I do think there has been a long-standing interest in being able to predict phenotype from genotype, so how a cell or an organism exists out in the world, given a sequence of letters.
Yeah.
And so certainly the increased data sets and computational power that we have now provide insights into how that could happen. I don't think we will really ever have a perfect prediction like that, right? So because we never ever measure some experiences of what this person has gone through in a way.
Right.
However, for certain changes like mutations in the BRCA1 gene, you can predict that the risk of breast cancer is fairly high. And so in those situations, you really do want to be able to think about medicine and prevention in a new way. I do think there's this story in the genome editing field that's really interesting about a gene called PCSK9.
Okay.
This is a gene that a company and several companies are trying to edit in the liver, and there are patients at the moment that are in clinical trials where a genome editor is being administered to the bloodstream in a nanoparticle that degrades, and it accumulates in the liver. It changes this gene in the liver of these patients, and it basically knocks out that gene. So that gene is not functional. Okay. And the reason they went after this target is that there were genetic studies of kind of human variation that found people would have a high-fat diet and they would never have cholesterol problems.
Wow.
And they traced it to these variants in PCSK9.
Okay.
And so that is a target that I think we could have widespread interest in having that is not necessarily therapeutic, but is important in certain environments and certain diets, and may not be required in worldwide, globally, but in some cases it's very helpful to have a PCSK9 inactivation.
Yeah, very good. What got you interested in this? This is a very deep and very arcane area of study. What got you interested in researching this? I've
always had an interest in thinking about controlling cells in a for therapy, and I had done a lot of research in my training on making new synthetic materials, so plastics-based polymers, and being able to tune how stiff it is or what other proteins would be attached to it. And I worked with stem cells, understood where this could go. Overall, I found that the effects were important and could control what stem cells could do for certain types of behaviors. But I was struck by how much more powerful changing the genome was. There was essentially two sets of discoveries that happened during my training that both led to the Nobel Prize after the fact, that led me to go into this direction. So the first one was this idea of taking an adult cell and reprogramming it to an embryonic state. So to that point, everyone thought that natural human development would take a cell, an embryonic cell, and you can only go one way towards an adult maturation.
Right. Time only moves in one direction.
So there was a set of studies. One scientist in Japan, Shinya Yamanaka, as well as work here in Madison by Jamie Thompson, was able to essentially take a skin cell from a patient and make it into an embryonic state. And so this was all done inside the genome, and that to me was really exciting. So I ended up doing quite a bit of work there. And then the other discovery was the discovery of CRISPR, which happened right when I started my lab here in Madison. And
that's the process.
CRISPR is the tool
Okay.
that we could use for genome editing.
Okay.
And so the first reports of this tool being used in human cells came out in 2012 when I started my lab. And I was in a perfect place to be able to test out whether that would work in the projects I had, and also to pivot in terms of using some earlier forms of genome editing to using CRISPR. And because it was so versatile and powerful, we ended up moving forward and doing some of the retinal work that I mentioned earlier.
Finally, well, two more questions, probably quick ones. One is there anything about your research that you would like our listeners to know, but for which I have not asked the right question?
This has been a great conversation.
Oh, good.
The answer might be no at the moment, yeah.
Okay, fair enough. No. And then the last thing I want to ask you is just when you're not involved in this heady research, what do you do in your spare time? Do you have hobby, sports, anything you like to do when you're not in the lab?
I'm a big basketball fan.
Oh, terrific.
And so most recently I was coaching my daughter's basketball team, which was a joy.
Nice.
Absolute joy. I get to talk about my time as a Chicago Bulls fan, which is
nice.
Different because I grew up in the South and knew about Milwaukee Bucks here.
Nice.
But Jordan certainly was on top of the world then. So
yeah.
I love basketball. I love the teamwork in it. I see quite a bit of parallel in terms of how I try to get cross-functional teams to move towards a common goal in science.
Yeah.
And I think there are really talented athletes that have contributed really unique skill sets to science. And I like that, thinking about that intersection.
Krishanu, thank you so much. This has been really enlightening. I know much more than I did going into this, and I'm excited about the potential. So thank you for the work that you and your lab mates are doing.
I appreciate it. This was really fun. Thank you.
You've been listening to the Badger Talks podcast. I hope you enjoyed this conversation with Dr. Krishanu Saha. Please browse our previous episodes for other topics that might be of interest to you. Badger Talks podcast is a creation of UW Connects and produced at Audio for the Arts Recording Studios in Madison, Wisconsin. Our music is composed by Bill Purdy and performed by the UW Marching Band. I'm Buzz Kemper. Thank you for listening.